Yes, welcome to the day 5 session on network pharmacology in internship and certificate course on computer aided drug and vaccine design. So today we'll be discussing on network pharmacology, system pharmacology, pathways, networks, interactome, diseasome, network visualizer, and case studies. Before going to discuss on today's sessions, as usual, can we recap the last session topics? So I just raise you few questions through polls. You can try to answer for that. I launched the first question. Identify the input file format of receptor and ligand in Autodoc. Just 30 seconds left out to answer this question. The choices provided are PDB, SDF, MOL2, and PDBQT. So, what are the input file formats? Just try to answer. If 20 participants are here, only one is answering. How about the others? Just come fast. We have just 15 seconds left out. 10 seconds. Just 5 seconds. Only 4 answered. How about the others? Try something. Yeah, I'm going to close the window soon. Can I close the poll? Yes, I am ending the poll. Only 5 votes I found. So the correct answer is PDPQT. You remember, yesterday I was discussing something on virtual screening methods. In virtual screening methods, I have provided you demo on Autodoc PYRX. You remember, in that I clearly mentioned that whatever file formats we have in ligand and receptor, finally we have to convert it to PDBQT format. Unless you convert, you can't able to go with docking. Okay, so the correct answer is PDBQT. Even if you have PDB, STF, MOL2, we have to convert into PDB finally, PDBQT finally. Then only we can go. Okay, now the second question I'm going to launch now. So the second question is, which is not the drug likeness filter? The choices provided are Lipinski, Mugi, Hans, and Weber. Which is not the drug likeness filter? So for this, I gave less time. Just 10 seconds left out. Still, only 5 are interacting. What happened to the others? 5 seconds left out. Yeah, I'm going to exit the poll. Try something. See, I'm not checking your responses. I don't know your names and email it is at present. If you want to answer, you can answer anything. Okay, I'm going to exit the poll, but this time I found 7 votes, 8 votes. Very good, nice try. So the answer is the majority you have answered. So the correct answer is hands. Okay, Lipinski, Milky, and Weber or drug likeness filters. Okay, so even you should recap the previous session topics. Okay, what exactly, and but if you watch the videos, these are very simple questions to answer. Unless you practice or watch the videos, it will be very easy. Okay, or else will be getting confused. Okay, now let me go ahead with the today's topics. Today topic we'll be discussing on these topics presently network pharmacology. You remember yesterday I just provided you some information on this. In network pharmacology, majorly we can find the networks, biological networks like protein-protein interaction networks, metabolic pathways. Okay, so these type of networks are generally used to come up with a kind of a network medicine. Whatever I'm telling, network medicine is a form of a precision medicine. So if you want to tailor the medicine according to a specific patient, we can able to make it using this network pharmacology. Okay, not only we identify one target gene which is responsible, but we also find multiple target genes followed by the interacting genes of the target genes. Okay, the whatever we tell like interacting genes of the main target, that we call it as hub gene. Okay, hub is the main gene, and the interacting genes are interacting genes. Okay, so we can find all this in this kind of a networks, and not only we find the metabolic networks and protein-protein networks, we can also discuss about disease networks. And yesterday I mentioned specifically on an expensive medicine called Zolgensma. So Zolgensma is actually manufactured by Novartis. This is used to treat spinal muscular atrophy. And in the spinal muscular atrophy, we have a target called human SMN protein. This is the target. For this, they use this kind of a gene delivery kind of shot. Okay, like a short vaccines shot, a single shot is around 1.5 ml used to treat spinal muscular atrophy. Now let me come across this network pharmacology. In network pharmacology, okay, what is the difference between the previous methods of drug design and here you see we come across multiple targets and of course multiple genes or proteins. In this case, if this network pharmacology is basically meant for fighting, combating the obesity. Okay, so for combating obesity, what kind of drugs we can use? Okay, for this, first we take the compounds responsible for obesity, and then we take targets and we find the corresponding genes and proteins, and then we find the PPI network, protein-protein interaction network for the particular hub protein, whatever we find. Then we go with gene ontology and check pathway analysis. Okay, and even we can able to find this kind of a network like compound protein networks. The compound protein target networks I told you we can find in Stitch TV. Okay, there is one database called String. The string is popularly meant for downloading the protein-protein interactions, and we can use the tools and this pathway analysis from Cake to get these kind of graphical charts. So whatever we complete this, okay, this is a part of a systems biology approach. System biology is a branch of science where we study many areas. One such area is network pharmacology. So one such area is the network pharmacology. Then what are the other areas we'll be discussing soon? Okay, so whatever we study this computational studies in a part of a system biology, we can also integrate with the in vitro studies. Like whatever we get, we can compare with the in vitro studies. That is testing the effect of these compounds on specific obesity cell lines. And if you want to check similar kind of experimentation in small animals like mice and rats, they can also, yeah. If you have doubts, you can ask someone else. Unmute. Do you have any doubts? If no doubts, let me take the next thing. Okay. So this is something like we can also compare in silico with in vitro and in vivo. Now this is another kind of a study. In this study, we take compounds like how we can able to work. In this pipeline you can see from the seeds or from the roots we take the active compounds, multiple compounds, whatever we get, we try to find the potential targets, not a single target, target 1 and target 2. Okay, so we can also go with the pathway analysis, and also we can check what are the other diseases associated on treatment, and then we can construct the networks based on our our problem statement. Systems pharmacology is the application of systems biology. So whatever we call as network pharmacology, we find a network of compounds, I mean network of proteins linked with the a compound. In system pharmacology, this is overall subject. In this overall subject, a part of a study is network pharmacology. So now we discuss more about systems biology. I told you, whatever we study network pharmacology, system pharmacology, this is a part of system biology. Then what is system biology? System biology is a branch of science that deals with types of omics like genomics, proteomics, metabolomics, transcriptomics, okay, interactomics. So whatever we study about the type of omics, okay, so these are what we call it as system biology. The purpose of system biology is to come up with a precision medicine. Okay, so for this we should also need networks, especially gene networks, and even the molecular structure, that is 3D structure of proteins, are also important. Okay, so the study of all this. So here in this we can see metabolism of all plus corporate metabolism, corporate metabolism, metabolism. So the metabolism, all we call it as system biology. In system biology can also be used to determine biomolecular computing. So if one area I told you network pharmacology, the second area is biomolecular computing. The biomolecular computing comes under cellular computing. Okay, so cellular computing is the general word. We use it even in IT, electronics, etc. In electronics, we call whatever we have a battery inside the cell phone. That is also a kind of a cellular computing. But in biotechnology, the cell is something different, isn't it? Whatever the organism possesses, okay, that's what we call it as cell. So under this, if you see, if you specifically say one specific type of cellular computing in biotechnology application, that is bacterial biomolecular computing. In this, we have this bacterial cell. This is like a CPU, and this is like an input and output, like a keyboard, and the output is something like a printer or monitor, something like that. Okay, you know, however, we find this biomolecular computing is looking in the form of a neuron. You know, in the neural network, we have multiple neurons. For each and every neuron, you have multiple inputs, and we can have one or more outputs. Similarly, if you see this kind of computational function, you know, the bacterial cell in computer is not exactly a bacteria, but we put a kind of a unit and we use some correlations, and using that correlations from whatever input we fed, input and weights, we take to correlate, and we for that we can able to generate outputs. Okay, this is what computational we do. And if you see this bacterial biomolecular computing, so based on the utilization of lactose, the intake of lactose and light, okay, some bacteria will exhibit the fluorescence. Example is Pseudomonas aeruginosa. Pseudomonas aeruginosa. It's very famous that it can also not only exhibit the fluorescence, but it can also uh, do the quorum sensing. Okay, and few bacterial cells can produce electric current, and based on the temperature and pH, some bacterial cells uh, can exhibit the pigmentation. So what you understand, so based on only the inputs, fed to the bacterial cell, we'll be getting these kind of outputs. Okay, this is what bacterial biomolecular computing. Of course, this comes under system biology. Generally, for doing this, we use E cell and B cell. These are the software tools generally we use for calculating the biomolecular computing. So instead of this biomolecular computing, that is instead of bacterial cell biomolecular computing, we can also able to work with molecule, like keeping a protein as a main processing unit, and we can give some inputs and outputs. Okay, and similarly, instead of gene and protein as a molecule, we can also work with organelle like a mitochondria. Instead of bacterial cell, we can have mitochondria. So how much ATP is given as an input, and how much outputs we'll be getting from this ATP. Okay, so all these things you can able to work with any organelles, not only mitochondria, chloroplast, during the photosynthesis, etc. And then cell, or whatever we discussed is a bacterial cell. Already we discussed. Similarly, we can try with the organism, etc. Now let me take you to the network biology. So network biology is a part of system biology, where we discuss this as it's a branch of science which involves biology, network theory. Whatever I'm telling network theory is a graph theory. In Max, there is one separate subject called graph theory. So uh, the network biologist should have a knowledge on basic biology and network theory or graph theory and statistics. In mathematics, one should know about statistics. Okay, so this is something about network biology. But in system biology, what exactly we discussed? We discussed the study of types of omics and gene networks. Okay, uh, gene networks and protein networks, etc. Okay. Now let me come across the main parts of network biology and what are the contemporary researches happening at present. You know, in this 1999 to 2005, much of the genome projects are established. If you see the human genome established in 2003, and if you see mice and rat established in 2001 and 2002, so most important small animals and human for doing experimentation on human diseases, okay, these genomes are established in this era. So the biological data came to existence and the more population happened in this 1990 to 2005. But before that, we have very less data, very less platform text data. And after 2005, what kind of evolution happened? So after genome, the scientists worked on to come up with a proteome databases. Proteome, especially protein-protein interaction. Only after 2005, many scientists take an initiative on this. And even much of the protein-protein interaction databases are established in 2015 itself. If 2015 the proteomes are established, then after 2015, many scientists try to focus on to produce interactome, metabolome, interactome, transcriptome, metabolome, transcriptome. What do you mean by transcriptome? It is RNA interactions. Okay, transcriptomes. But whereas protein interactions, genome, we can find the genes. Now, if you see this kind of era, this is what the main thing, whatever happening in contemporary research in computational biology. Now, let me discuss something on networks. What are the types of biological networks we can see? So, the most widely used networks in pathway analysis is protein-protein interaction network. We also have protein DNA interaction network, gene correlation, gene regulatory, metabolic signal. Okay, so these are the type of networks you can see in the databases. For example, if you see the metabolic network, the best example for metabolic network is glycolysis, Krebs cycle, whatever we discussed Krebs cycle, whatever we discussed in biochemistry, we have studied in biochemistry. So all these things comes under that type of metabolic network. You know, in glycolysis, how we start? We start with glucose, and after 10 steps, we try to get pyruvate. Meanwhile, you can see multiple intermediate steps. isn't it? So whatever metabolites formed in between this, okay, can able to see this metabolic network. Similarly, in Krebs cycle and other cycles. The next thing is protein interaction network. This is an example of yeast protein interaction network. If you see the yeast, okay, we have more than 99 percentage of genes code for proteins. So if genes code for proteins, okay, then it means that we have now more than 99 percentage exons. If this is the case in yeast, if you compare with the human genome, I mean, if you compare with the human, okay, you can see we have less than 2 percentage of genes code for proteins. You see in yeast more than 99 percentage code for proteins, but in human we have less than 2 percentage code for proteins. So we have very less exons. Whatever we call it as non-coding genes are present more in humans. But if you see this kind of a picture, this is a yeast protein interaction network. A single unicellular organism has multiple proteins connected in the network, and this is a gene regulatory network. In the gene regulatory network, we can find two different colors. One is red color, the other is black color. And whatever we find here, these are genes. For example, P10 gene, CCP1 gene. There are so many types of genes. And the red color, once again, I'm telling, this is positive regulation. If red color is positive regulation, the black color is negative regulation. So if you see the main gene called TP53, the TP53 is most important gene for breast cancer, cervical cancer, most of the cancers. This is ranking number one, most important gene. So if you see this TP53 interacting genes, if TP53 is considered as a hub gene, and if you're finding a top 10 interacting genes with TP53, okay, some genes can positively regulate, some genes are negatively regulate. Positive genes are shown in red color lines. The red color lines, whatever we see, we call that as edges. Lines is what we call it as edges. So positive edges are in red color and negative edges are in black color. In this gene regulatory network. Gene regulatory network is different. Gene interaction network is different. In gene interaction network, we find in only one color. Okay, either black or red. But only in gene regulatory network, you have two different colors and two different distinguished genes can be found out from this. Now, in this slide, we discuss on how a small graph is constructed. So, if you have this, okay, we consider if it is a protein network, this is protein 1, this is protein 2, this is protein 3. Okay, if it is a gene interaction network, this is gene 1, gene 2, gene 3. So, if you want to form a small kind of a graph or network, if you have two proteins like P1 and P2, okay, so whatever I draw this from central point of this protein to this protein, is this we call as a graph? No, we can't call it as a graph. Then what is it? This is just a line interconnecting protein 1 and protein 1 and protein 2. But here, just because you find in the form of a cyclic path, P1 is connected to P2, P2 is connected to P3, P3 is once again connected back to P1. Just because it's following a cyclic path, then only now we call this as a graph. For a simple graph, we should have minimum of 3 nodes. According to the graph, whatever the dots I represent, that's what we call it as nodes. If this is called node or vertex, whatever connecting the nodes is what we call it as edges. So this is actually edge. Okay, but we should have one more protein to connect in it. And see, one more thing you can infer from here, that if you see this P1 to P3, how is it connected? You see, there are 3 paths available: path A, path B, and path C. So, which is the best path? Obviously, the best path is The shortest distance. The shortest distance of when you compare A, B, and C, the B is the best path. Okay, shortest path. But some proteins or genes, instead of following the shortest path for connection, it can also use either path A or path C. Okay, so if you see the path C, it is you find the edge distance is more. So this is not generally preferred, but sometimes it also follows these kind of path. So the next to the B path, the A path is the best one because it shows a smaller distance when compared to C. Okay, this is something about graph theory. Now let me take you to the further uh, biological networks. So if you want to come up with a kind of a precision medicine, initially we have to work on genome. Then only we have to work on proteome. After working on genome proteome, in a pipeline we have to work on metabolome. Got it? This is how we work. In metabolome, we can find even the biochemical reactions and the metabolites formed in that reaction. Okay. So all these things have to be integrated to come up with a precision medicine. Example of his protein interaction network by Hawang Cham. Gene regulation network of CR gene. So you can see different colors of positive regulation, negative regulation. Metabolic flux analysis of E. coli. Okay. This is a glucuronate dissimilation. You can see pathways like gluconeogenesis, oxidative phosphorylation, pentose phosphate phosphorylation, fermentation. All are interlinked. Nearly somewhere we find pathway 1, pathway 2, pathway 3, pathway 4, pathway 5, pathway 6, pathway 7. 7 pathways are interconnected here in this metabolic flux analysis. Now we can see another picture similar to that. If you see, just now I mentioned, am I audible to everyone? Yes or no? Yes? No. Participants, I in the
Yes,
chat.
sir.
Yes, yes, okay. Yeah, yeah. Okay, okay. Thank you, thank you. See, just in fact, this is most important. I don't know whether I'm in line. Thank you for the response. Now let me go ahead further. So if you see here, glycolysis. Okay, we start with glucose. This is the input, no doubt, and the output what we get is the pyruvate. But the output of pyruvate, I mean output of glycolysis, which is a pyruvate, can be input of TCA cycle. Okay, and you get a different kind of an output. And similarly, the pyruvate can also be working like an input for ED pathway and for pentose phosphate pathway. So all these pathways are interconnected with each other based on the metabolites produced. Okay, so this is what extracted from KIC metabolic pathways, linking all major pathways. Pathway role in target identification. Okay, so generally, if you see to work on pathways, we can also use these databases like Impact. In Impact, we already have a network database. Can I show you something directly to these websites? What exactly is there here? So first we go to the Impact. So if I click this Impact, I told you this is the largest Indian medicinal database. If you see this Impact, Indian medicinal plants, phytochemistry and therapeutics, we can able to search data either through phytochemical or therapeutic use. So not only this, if you drag down, you can find this kind of a window. Here also you can search. For example, based on therapeutic use, I want to search the plants which is best. Okay, so there are multiple diseases mentioned here. You can able to specify, select any specific thing. For example, asthma. If I click asthma, you'll be getting these kind of medicinal plants. You know, around 1047 plants are used for treatment of asthma. Not 1047. Actually, whatever we found that 1047 is at different parts, including the different species. For example, Apras precatorius fruit can be used for treatment of asthma. Similarly, leaf. Same plant, the leaf can also be used for treatment of asthma. So you find the species, genus and species name, and also the parts of the plant. Okay. So together we find nearly 1047. And the therapeutic use of all these plants and the part plants, parts of the plants is to treat only the asthma. Now, if I want to, if I click this asthma, okay, so I'll be clicking this asthma. All right, I just click this plant presently. So if I click this plant, okay, so you can able to see this plant is also is not just limiting in treatment of asthma, but it's also used for treatment of wound healing, wound injuries. So what is this? This is a kind of a network created. Okay. Similarly, you can search for any diseases. Okay, a plant linked with not only linked with one particular disease, you can also find multiple diseases. Okay, like however we find, okay, you can search with any specific plant. Okay, we find multiple diseases or this specific caliber indica is treating multiple diseases which you can able to see in the plant, right? Okay, so like this you can find the next database is the Hub 2.0. So for Hub 2.0, this is the hyperlink. If you type this hyperlink, this takes you to the Chinese traditional medicine database. So this is the IP address for that. Here also you can find much of the network information. So it's currently loading, but meanwhile I can show you the PPT. So in therapeutic use, I selected a particular application or if I in the plant. If I click the plant, obviously I got this network like this. Okay, this is what we have seen just now. And now Herb 2.0. So in Herb 2.0, so you have to provide an input of a plant compound. If I provide an input of a compound like vitamin A, the vitamin A is interacting with these proteins. Whatever we find here, these are the interacting genes or proteins. Okay, so this is something different. Earlier we had a different network. This is something different network from Chinese traditional medicine database. And we can also see how many genes are interconnected with each other. Okay, this is a statistical result. The next thing what I'm going to take you is network analyst. In network analyst, we can able to see the change in interaction networks. So this is a comprehensive gene expression profiling. Okay, this is a comprehensive gene expression profiling. When we can go with the gene expression profiling, especially if you want to treat cancer with precision medicine, we need these kind of a gene expression profiling. So how this can be done? Via network visual analytics. To create the networks, we can use this network analyst. Okay, so this is something about how the file of gene expression can be used in network analyst. Now, in the network analyst, we also have one module called Express Analyst R. In Express Analyst R, we can able to do the gene expression controlling through R programming. R programming is one of the important programming used for statistics. Like if you want to generate graphs, okay, so types of graphs, R programming is the best. And whatever gene expression data we have in network analyst, if you want to show it in different graphs, we can link with this R programming, okay, which is already present in this network analyst that they named as Express Analyst app. So download the sample files and you just execute in the same kind of tool present in network analyst through R program. So in Express Analyst tab, we have these kind of a windows. So library Express Analyst tab, we are exporting this kind of a library, and we have plenty of functions to work on gene expression. Now we can see one graph at the bottom. The gene expression is so shown in this kind of a plot. Okay, the bar plot. So how is this done? This is done only through the. This is actually box plot, not plot. This is what we call it as box plot. So the box plot is done through express analyzed through R program. Okay, so this is how we can use this as a database and tool to analyze the gene expression patterns. The next thing what we discuss is the metaboanalyst. In metaboanalyst, we have different metabolites, and these metabolites in the network can be analyzed here. Okay, how we can get the metabolites? Metabolites we can also get from uh, multi spectrometry, multi T wave spectrometry, like that. Okay, so whatever whatever metabolites we produce from these wet lab experiments, these are deposited in this metabolite. That is, metaboanalyst is used to measure the analytics of this metabolites present in the database. So as I mentioned, so we can also use LCMS. So through LCMS, so much data is deposited here. We can use this metaboanalyst R, okay, which to work with the metabolite. The next thing what we discuss is the DGNet. A DGNet is a disease gene networks. Okay, if you see the DG network, so actually in the next slides we have. Okay, I take you to the website. Uh, So I was explaining you something on Herb 2.0, isn't it? So if you focus on a specific ingredient, for example, Vitaferin, I'm typing the Vitaferin, you're getting the types of Vitaferin also. Okay, so if you go with this, you can able to find her. Okay, so this is a kind of a network. We find the network of compound with the diseases. Okay, we have different hyperlinks here, related targets. So if we click this, we find the targets which they can able to find. And here this is also linked with the DGNet database. For each and every disease we have ID. Okay, so if you click this ID, disease ID, so we get this information of disease name, alcoholic intoxication, chronic. Okay, so for this you can find this information. So if you see this kind of a network, okay, direction of connectivity mapping. So this is the disease name, alcoholic, whatever mentioned. So for this, what herb and how it can be used for the treatment. Okay, so we can find this kind of connectivity mapping here. We also find the networks with the compounds present in the specific herb. Okay, table data is there, genes information is there. So if you drag down, you can find plenty of information. Patient analysis. Okay, so this is something about Herb 2.0. Okay, also linked with the Digi network. But if you want to go exclusively through Digi network, type Digi net. So if you see the Digi net, you can find either you can search through genes, diseases. For example, if I want to find through EGFR, okay, a gene. You can also type examples like obesity and other disease names. Instead of disease names, I type just like EGFR. The EGFR is linked with so many, is linked with diseases, genes, variants, what exactly we want. I'm searching only with the genes, isn't it? So if you want to explore on this, you select this search. So whatever EGFR gene information, if you see, this is linked with this much number of diseases. Carcinoma is the non-small lung carcinoma, squamous. So these are the types of genes. Okay, and you can see this one particular gene is causing this How many results we got around? We got around 2648 results. One gene EGFR is responsible to cause around 2648 into number of this. So totally in a thing, if you have a 10 results, 10 into 2648. No, totally 2648 results we have. So like this, you can search for a particular gene and you can check the gene is responsible for what type of diseases. Okay. On 23rd, you can also see how one disease is linked with another disease. For example, the cervical cancer patients are more prone to which viral infections? Can you someone tell me? This is my question now. Cervical cancer patients ha- have more prone to what viral infections? There is a relationship, a viral etiology with respect to cancer. You know, the answer is HPV, human papillomavirus. Human papillomavirus infections, if there are 99 percentage of HPV infections, definitely they'll be getting cervical cancer. If you see the cervical cancer, definitely they have 99% HPV infections. Okay, so like this, you see a few cancers are caused just because of the viral infections. So for this kind of net can give you some more information on that. Okay, idea on that. Okay, now let me go ahead with the next database. The next database, whatever I'm going to show you, even in the G network, there are much of information to explain. For example, if I go with a non-lung, small lung cancer carcinoma, we can get something like for EGFR, we got disease DPI, DSI. Okay, what is DPI? What is DSI? So all these things will be seen in our PPT. If you see in disease zone, okay, that is the GNet is majorly used for studying disease zones, that is human disease networks. Okay, the metabolic disease linked with another metabolic disease in the pathways. If you see in disease zone, there are three representations. Shared gene formalism. That is, if a gene is linked to two different disease phenotypes, then the two diseases likely to have a common genetic origin. This is what shared gene formalism. And shared metabolic pathway formalism. That is, if a metabolic pathway is linked to two different diseases. For example, NF kappa B, kappa beta pathway. Definitely, this NF kappa B is linked with ovarian cancer, cervical cancer, and other types of cancer. So this pathway related information, if for this also I can find it. This is okay. For this, they are mentioning they have a shared metabolic origin, and similar disease comorbidity formalism uses phenotypic disease networks, abbreviated as PDN, where two diseases are linked if observed comorbidity between the phenotypes exceeds a predefined threshold. And if you see in disease gene network, okay, we can also integrate with the R package, R programming. And then we can also link with the, if we want to visualize the disease networks or genes linked with the disease, we can open it using the cytoscape visualizer. You know, the cytoscape is used to visualize any type of networks. Can be disease networks, gene interaction network, gene regulation network, protein network, DNA protein networks, any type of proteins type of networks can be visualized using cytoscape. The JNet, we directly link with the cytoscape to visualize all the networks. We also can customize our scripts using Python programming, R, Perl programming, also link with the R, okay, to come up with the various results, graphical results. And the GNet, you know, you can also search much of the information in online related to genes and other diseases, how it is connected. So when I just gone to the GNet network, I got something like DSI. Remember, DSI is disease specificity index. For example, there are genes or variants that are associated with multiple tumor necrosis factor. You know, the tumor necrosis factor is generally used for combating cancer. But the same protein, tumor necrosis factor, if present in excess, this can lead to autoimmune diseases. Autoimmune diseases can be psoriasis, asthma, arthritis, whatever we discussed on yesterday. So the TNF should be in regulatory level. It should not be excess, it should not be less. Got it? So, if a particular protein we take and if we study with multiple diseases, we can measure this based on, like, if TNF is linked with 15 diseases. Okay, so based on that 15 diseases, we can able to use the formula. So, for example, they are mentioning if TNF is associated with 3,500 diseases, not 15, we got around this linked with 3,500 under this case, so we got like if the DSI is ranging from 0.23 to 1, the DSI has 0.3, so it has a least value if it is connected with much of the diseases. But if one gene, for example, one particular gene is responsible to cause only one disease, for example, MOP3 is associated with one disease. Under this case, we get a range of disease specificity index of 1. It is connected with multiple diseases. This is the lowest value point, so that you can able to find it for TNF. But for this, we got as well. So similarly, if you check the disease pleiotropy index, in the DigiNet, we generally get the results. But how to interpret the results, how to infer the results, that's most important. So according to this disease pleiotropy, you see, according to this, if the multiple diseases are associated to the gene, for example, EGFR, TP53, this gene is linked with multiple cancers. So under this case, we can able to measure this kind of pleiotropy through disease pleiotropy index. Okay, this is the formula, and examples are mentioned. Okay, if you see this kind of an example, okay, for elephant, you see whether the elephant can able to get any type of cancer. So this is a general question I'm asking you. Did you ever heard that elephant can able to get cancer? Yes or no? Elephant can get cancer or not? I think I'm audible to everyone. I'm just asking you. The humans are getting all types of cancer, isn't it? If you take that oral cancer itself, we have tongue cancer, tooth cancer, lung, everything. Okay, lip cancer. We have all the types of cancers in human. I asked you a question that elephant can get cancer. No, sir. Chances are very less. Your answer is perfect. You know what is the reason why the elephant is not going to get cancer? Because the elephant has multiple copies of TP53 gene. We have only one gene. Few few humans lack even the TP53 gene. The TP53 gene is important to combat cancer. Okay, so elephant won't get any type of cancer because it has multiple copies of TP. So the genes are playing a major role to cause the disease. There is also one more kind of a measuring indices called evidence disease evidence index. Okay, so like this you see, if I take you to this Tichy network, okay, you find simply this DPI, PSI, like this. The values are mentioned. But if you want to know more about this, you can see the literature review and then you can finalize. Uh, how about the influence of gene in a particular disease? Got it? That's what we call it as the gene network. Now, let me take you to further. What are the other results in the gene network? Already we discussed. Now I take you to the cytoscape. Cytoscape is an open source, that is, it's a freeware. So anyone can able to install because even it's going to occupy very less space, and this is used to visualize any type of networks. So if you see the input features of network, okay, it can be DNA, protein. Okay, so anything, we generally upload the kind of files in the form of Excel sheet. So generally .tsv tabulated separate value file which we can able to open in Excel. This can be provided as an input for cytoscape. And if you see how the nodes are connected to the network, so the nodes are connected to the network, okay, through graph modeling language. So the corresponding files will be in GML format. We also have extensible graph markup and modeling language formats. The network language is something different. We have different languages. Only through the languages we can able to build the networks. Can be mRNA interaction networks, DNA networks, anything like that. Input features of cytoscape. Okay, so I told you input features generally we can able to open any kind of DSV file which we generally open in Excel sheet for editing the data. If you see in Cytoscape, we have plenty of apps to work on various applications. I told you in network analyst, we majorly work on gene expression analysis to check whether the patient is in late stage of cancer or initial stage of cancer. So if you see in Cytoscape, we are not limiting up to the gene expression networks, but we can also use it for multiple applications, including reactomes, protein networks, etc. Okay, if you see the apps for each and every app, we have a distinguished icon. So any kind of a data from these databases like the G network, INOM, NBIC. So whatever we find here, this can be visualized using Cytoscape. So we also have plugins or apps. If you install that, it can work with all these tools. So in the Cytoscape, I just visualize the yeast gene interactions. Okay, it looks like this in Cytoscape. And this is an example of visualizing TCGA, colorectal cancer. So here you can see proteins are interconnected with each other. Now let me take you to the case study one. So before going to the case study one, I just teach you like how to use the string DB. So I believe I already taken, but still just recapping once more. If I take you to the string DB, which is the database for protein association networks. Okay, if you have to simply type the protein name. Last time you remember, I typed trap1. This is heat shock protein 75. When I searched, okay, I'm getting you have not provided any. Go back. I just provided search. Can I type something else? HSP90A1. There is actually some problem with the string database. This is how we have to search. The server is down. That's the reason. Okay, then I can take it to the next database called Fund2. I believe in the first day I explained about the string database. Now this is another database called Fund2. You can see change in interaction, protein in interactions, etc. Like however is searched, here also we can search. Now I'm going to type like HSP. I'm getting anything, or I type simply trap. Search for homosapiens. So we're getting this trap one. Whether you want to find this kind. No, I don't want this. I want each of in sample. So selected this. Continue with search. So there are two identifiers for two types of proteins. The identifiers are same. So I'm selecting the one which I'm going to show. So this is a fun book database presently going to show with protein-protein interaction networks. So interactions, chapters, enrichment, everything you can find there. So the network is getting loaded here. Meanwhile, I can take it to the Stitch database. Stitch TV. If you type default whatever we get, Stitch database notes. Not this notes, the first one. Still it's going to somewhere else. Stitch EMPL middle bar stitch. So here you can search through protein. It's asking like enter an amino acid sequence. Instead you can go with item name, tab one. The search you can find protein interactions. Also you can check the ligand protein interactions in the stitch database. So trap one is interacting with these proteins. If you want to increase the connections, so instead of no more than 10 interactors, put the custom value type 100, update. You can find the number of interactions will be increased. So it's executing at present. When you find the output, you can find it. This change still standing here. The network is slow actually. You'll be finding a network here. It's not at all. Here still it's not. Can I change? So, can see this gene expressions, patterns, PPI, score, etc. Oh, observe is presently the server is down. Not the internet. The server is many are accessing at the same time. Again, I can't make this. We are getting this. But settings, replace the interactions. Update. If server is slow, then I can take another next step. Here also have to get a type of protein. However, you find in this, okay, in SysDB, even in front, yeah, now we got this. This is the protein interaction network of the specific protein item. Okay, so you can find this. You can also increase, you can find the interactors information, enrichment information, how you want to enrich the genes. Whether whatever target protein, hit shot protein 75 I type, whether you want to work on cervical cancer, breast cancer. So all these things you can able to do the enrichment using the pathways and other gene ontology functions. So by selecting few kind of information, you can enrich the genes. That is what gene is majorly interacting with the trap. So like this, you can able to find. So you have some problems. Okay. So now simply I'm going to it's lost. Okay, so we can also download the protein interactions. Okay, you can also download the protein. I'm going to close the other windows. So there is a visualizer called Cytoscape. I can download one file from the stitch or string database. Then I can show you how. Now I check whether it's opening. This is a peak time system. So this this is open 100%. If we want to download this, go to the tables. We have TSV file here. In the tables, we have TSV. Just select this. Oh, because of support. Okay, so if we have a good kind of if there is no such a server kind of a traffic, then I can show you all this information on tomorrow. Okay, so today I showed you something like how to visualize the networks. But earlier I've saved this kind of networks. See, if I select 100 kind of a proteins, you get like this kind of a file, .tsv file. If you open with the cytoscape is a visualizer. So the top 50 genes I downloaded in another file. Now I'm showing you the top 100 genes from string database. The same thing, if you want to edit the file, you can visualize it using Excel. So Excel is not presented. So I just open the Excel. I'm going to. So I just opened an empty Excel. If you want to visualize this data, I just drag and drop. You can find this. This is the data actually. This is the data of Dragon interacting with 100 proteins. Okay, so we find plenty of data here. If this is the editable data, if I open in the cytoscape, okay, that 100 interacting proteins, this is the visualizer. Actually, it's asking to download a recent version. There is no need, simply close. If I want to open the data table, you click this and open this 100 tab. So now you find this kind of a table like whatever we find in Excel. Simply we have to click OK. Simply we have to click OK. So the network is going to be loaded soon. Yeah, the network is loaded. I have used this kind of an option to load the protein. So now if you want to select this and if you want to color, if you want to do the style, you can fill with a different color instead of blue color, whatever you want to fill. Okay, like for example, you want to fill with this color, you can select this color. Okay, and then click OK. So this alone will be in red color. So we can able to change the colors, we can change the font, we can also find the gene enrichment. Based on the gene enrichment, what are the most important genes, that alone you can color accordingly. Okay, so if you want to know more about the cytoscape and how to use, we have help user manual. It gives you detailed information on how to use different apps, etc. And even we have tutorials, we have tour, we have video demos, excellent demos in YouTube. Multiple video demos are there to how to use the cytoscape. Okay, and other plugins present in cytoscape. So anyone can able to learn by using this help option present in cytoscape. So that's it. So today I have completed something on network pharmacology. I've shown you the types of networks on today, and tomorrow we'll be discussing on molecular modeling, that is small molecule modeling on Saturday. And Sunday we have a break. Okay. Any doubts you can ask me at first. Do you have any doubts? If no doubts, thank you very much for patience and interaction. I suggest everyone to practice. Okay, so whatever video I share, you practice, then you get more knowledge and you can work on even the projects. Publication of paper is not a easier task if you know the computational biology. You can reduce the time and money if you learn these skills and spend time on publication of research papers in the journals. So thank you. Have a great day. Good night.